This is Psalm 9394. My name is Karen. In today's episode, I wanted to look at some of the updates with technology which have appeared in the news this week. First up is an article from Yahoo Finance called DNA Nanobots Gain Exclusive License to UC Berkeley Gene Editing Technology Platform for Non-Viral Gene Delivery Solution using DNA Nanoparticles and CRISPR-Cas. Biotechnology leader in DNA nanoparticles called DNA Nanobots announced it completed exclusive commercial licensing of a gene editing technology from UC Berkeley. This new licensed intellectual property enables the folding of genes into their own delivery devices, and this is working with the CRISPR-Cas system. The doctor and first author of the research study is quoted as saying, "I'm thrilled to see technology for large gene delivery and replacement that I co-invented and co-developed during my time at Dwadna Lab to continue to advance DNA nanobots. I'm excited about the potential of DNA nanoparticles in genetic medicines, given their ability to simultaneously encode the gene of interest while also being programmed and designed as novel delivery vehicles." If you'd like to learn more about their technology, you can visit dnananobots.com. This is a bit technical, but their website says what we do. We provide solutions, transforming payloads into precision medicine. We specialize in crafting custom-targeted therapeutics delivery using our DNA nanoparticle platform. Their website also says DNA Nanobots is helping companies deliver their treatments while developing their own. Reaching out to companies, they say, "We'd love to partner our delivery technology to your chemotherapy drug, siRNA, protein, gene, or peptide." siRNA is a small interfering RNA, sometimes known as short interfering RNA or silencing RNA, in a class of double-stranded RNA, at first non-coding RNA molecules. DNA Nanobots' website says they believe with the right tools and expertise, they can unlock the potential of custom DNA nanostructures and create powerful solutions to some of humanity's most pressing challenges. Their products make it easier than ever to assemble complex DNA nanobots with unprecedented precision. Just remember, this is what would be happening inside a person's body. The senior research scientist from that company was recognized with the prestigious Pelotonia Fellowship in 2020. This is based on something called DNA origami from the National Human Genome Research Institute. They say you may recognize DNA as one of the most well-known biological structures, but what better way to understand the actual twists, turns, and rules of base pairing than to make your own DNA origami model? The most common shape of DNA in living cells is a right-handed double helix called bDNA. DNA can also come in two other shapes. aDNA is also right-handed helical structure, but it is shorter and wider and found in dehydrated samples of DNA. Another one is called zDNA. It's an unusual version where DNA takes a left-handed shape. They say the left-handed shape is a temporary phase existing in response to certain cellular activities, such as when certain genes are being transcribed into proteins. Origami has been used for religious ceremonies at Shinto shrines. The word origami is derived from two Japanese words, ori, meaning folded, and kami, meaning paper. In other technology news, gene editing's next big targets. This appeared in Axios. A virtual event was hosted by Genetic Engineering and Biotechnology News. They said gene integration is one approach being hotly pursued to treat certain diseases in which cells don't make enough of a certain protein. This involves splicing the correct DNA sequence into the genome so more of the protein is produced. According to Jonathan Gutenberg, a Harvard Medical School professor and co-founder of Tome Biosciences, a biotech company focused on gene integration technologies, this is the last big bottleneck which a lot of people are working on. Another area scientists are working on is RNA editing and another frontier. This will allow larger edits to be made without altering the patient's DNA. The goal is to do corrections at the RNA level without ever having to touch the genome, Mr. Gutenberg said. According to the article, gene editing's biggest successes so far have largely been around inherited disorders. So what's next? Beyond humans, gene editing is also being used for precision breeding of cattle, pigs, and other livestock. According to the scientist, unlike genetically modified organisms that receive DNA from other species, gene editing involves altering the animal's own genome to yield changes that scientists say could arise naturally through selection. There's a company called Genus, and that's spelled Genus. They're working on making pigs resistant to disease. That company is seeking FDA approval for widespread consumption of its gene-edited pigs. Under the What to Watch section, which they covered, it says the FDA last year authorized pork from 5 gene-edited pigs to enter the human food supply in a limited way. Isaiah 66:16 says, "For by fire and by his sword will the Lord plead with all flesh, and the slain of the Lord shall be many. They that sanctify themselves and purify themselves in the gardens behind one tree in the midst, eating swine's flesh, and the abomination and the mouse shall be consumed together, says the Lord." If you look in the Blue Letter Bible, it shows that the word tree has been inserted. So that should actually be read as they that sanctify themselves and purify themselves in the garden behind one in the midst, eating swine's flesh, and the abomination and the mouse shall be consumed together, says the Lord. If you saw a video on my Rumble channel called Isaiah 66 Mouse Swine Abomination, we looked at an article titled Canadian Researchers Create New Form of Cultivated Meat. Basically what they do is take animal cells and put it in a stainless steel vat, and it begins to grow much like a cancer cell would grow. This article is dated 2021, but the similarities with the origami pattern developing are very similar. It says the sheets of living cell, each about the thickness of a sheet of printed paper, are first grown in culture and then concentrated on growth plates before being peeled off and stacked or folded together. The sheets naturally bond to one another before the cells die. They say, quote, "We are creating slabs of meat." As they describe in the journal Cell Tissue Organs, the researchers proved the concept by making meat from available lines of mouse cells. Though they did not eat the mouse meat described in the research paper, they later made and cooked a sample of meat they created from rabbit cells. They say there's no reason to think the same technology would not work for growing beef, pork, or chicken, and the model would lend itself well to large-scale production. The researchers were inspired by the, quote, meat supply crisis, in which worldwide demand is growing while current meat consumption is straining land and water resources and generating troubling levels of greenhouse gases. Meat production right now is not sustainable. There has to be an alternative way of creating meat. So what can we do? Use the Bible as our guide. Pray over your food and your drink and be very careful what you're putting into your body. If you found that technology bite helpful, please let me know. I hope that you have a great day and thanks for listening.